Today we're sharing with you some results of our research at the University of Iowa on how exercise enhances activity in the human brain. Even one session of exercise is known to acutely improve learning and memory in humans. Animal studies have offered cellular and synaptic accounts of these effects. In contrast, human data from neuroimage studies offers only a macro-level view of exercise brain effects. In the middle, the neurophysiological mechanisms by which exercise influences cognition remain unknown. Hippocampal function relies on a variety of electrophysiological events, the best known of which are ripples. Ripples are high-frequency oscillations that can be only detected with invasive recordings. Ripples play a critical role in memory. Replay of previous experiences happens mainly during these oscillatory events. Ripples are reduced with aging and altered in animal models of Alzheimer's disease. Recent studies show that the coupling between ripples in the hippocampus and neocortex reflects acute modulations in interregional connectivity required by mnemonic processes. Does exercise modulate hippocampal ripple dynamics in order to exert its effects on memory and learning? We hypothesize that exercise acutely increases ripple rate in the hippocampus and enhances hippocampo-cortical interactions in the ripple frequency band. Participants were 14 neurosurgical patients monitored for epileptic seizure localization with chronic intracranial electroencephalography recordings. Patients completed an exercise paradigm while neural activity was continuously monitored. The paradigm consisted of a pre-exercise resting state period of 20 minutes, followed by a period of guided exercise on a pedal exerciser, and finally by a post-exercise resting state period of approximately 20 minutes. Signals from 2,415 recording sites were collected from the 14 patients. Cortical recording sites were grouped into 7 canonical networks. We evaluated 3 aspects of the effect on exercise on hippocampal ripple dynamics. 1. The properties of ripples in the hippocampus. 2. Hippocampal-cortical ripple coupling. And 3. Hippocampal-cortical ripple phase synchrony. To test our specific hypotheses at the group level, we used linear mixed effect models with subject and recording site represented as random effects. In this way, we accounted for both participant-level variability and heterogeneity in electrode coverage across subjects. Ripple properties. The rate of hippocampal ripples and their electrophysiological features are known to vary across behavioral states and to predict memory performance. We found a statistically significant increase of ripple rate in the hippocampus after exercise. We then investigated whether exercise also modulated the electrophysiological features of ripple events in other cortical areas. Cortical sites were grouped according to 7 canonical cortical networks. We found that exercise increased ripple event rate and duration in the limbic network. Notably, the default mode network exhibited significant increases in ripple rate, duration, and peak frequency after exercise. Ripple coupling. Ripples in mesial areas often co-occur with similar high-frequency events in other brain areas during pre- and post-exercise resting state. This phenomenon is referred as hippocampal-cortical ripple coupling. To quantify ripple coupling, we performed cross-correlation analyses between ripple timestamps. Group-level analyses showed that, after exercise, ripple coupling increases predominantly between the hippocampus and the limbic network, and between the hippocampus and DMN. The ventral attention network exhibits enhancement in coupling with hippocampal ripples that reached statistical significance. Ripple phase synchrony. Recent studies have shown that ripples are phase-locked across distinct, yet functionally connected brain areas. Given the increase in ripple coupling we observed after exercise, we explored whether exercise also modulated phase synchronization. The phase synchrony between hippocampal and cortical ripples was measured by computing an amplitude-weighted phase-locking value. We found that after exercise, ripple phase synchrony increases predominantly between the hippocampus and the limbic network. The effects on the default mode network and front parietal networks also reached statistical significance. Overall, we found that even one session of exercise increases the number of ripples in the hippocampus. Moreover, the ripple rate and duration increase in the limbic and default mode networks after exercise. When we looked at the coupling and synchrony between ripples in the hippocampus and other regions of the brain, we found that neurophysiological connectivity increased in the limbic and default mode networks. In summary, even after one session of exercise, there is an enhancement of ripple hippocampal-cortical dynamics.